Hello students, welcome to lecture 3 of week 6. So we have seen that we need long read and short read data that we generate for various genomes and which we require for the assembly, right? So today we will discuss that what is long read data, how it is generated, which technologies are useful. Although we have already done a kind of detailed overview of the Nanopore and the PacBio sequencing technologies. So in today's class, we will have a quick recap of those and also see that how exactly those are run, for the various genome sequencing and the assembly algorithm that we use for their assembly, and also see some of the case studies where the data were produced by these approaches and what is the net outcome, right? So basically, Nanopore or PacBio are the third generation sequencing technologies, which provide long read data. The data like generally have errors up to 5% and may not be useful directly, but it also has some provision to remove the errors on its own. And later on, when you do the polishing using the error correction polishing using the short reads, that at that time also you can also make it like better quality, right? So basically it is of two types, the SMRT PacBio and the Oxford Nanopore technologies. So this is Nanopore based and this is a SMRT based technology, which you will see also recap today. Synthetic long reads are also there for third generation and similarly 10x genomics. So but this we have already discussed, and for this we have a separate lecture, right? So these two we will discuss that how they are produced and what are the assembly methods. So their advantages are long read length, because that is essential for resolving the repeats and other complexities of the genome. Haplotypes also, like polyploid in case of may have lower cost of sequencing. For example, the Oxford Nanopore may be lower cost as compared to the SMRT or PacBio, right? Sample preparation can be done in the lab and you can run it like easily, quickly, right? So like in case of ONT, you can also reuse the flow cell a couple of times, right? So basically the instruments could be PacBio, MinION, GridION, Chromium 10x genomics that we have discussed separately. So these are some of the instruments that can be used for third generation sequencing. So coming to the Nanopore or Oxford Nanopore technology, or Nanopore sequencing. So basically it deals with a nanopore, which is like this, from which the DNA, single stranded DNA passes. And that single stranded DNA is having A, T, G, C, 4 nucleotides, and which have their own shape, right? And basically the area or size that they are covering. So basically in the nanopore there is a continuous flow of ions. So right, so basically when there is something like a DNA which is having a different shapes of different nucleotides, they will block this pore to variable extent, and that will lead to disruption in the ionic current or voltage that is detected, and that will be used to detect that what molecule of DNA like A, T, G, C pass through this, right? So these are protein pores embedded in a membrane. You can do almost 400 bases per second in real time, so that you can, when it is passing through, and after that you can quickly see that what bases you have produced. So that also helps you to quickly correct if there is some error, or also allow some of the pores to do sequencing and some not, so that you can do selected sequencing also, right? So it's real time data streaming, and it eliminates the PCR bias that is that may happen in case of the Illumina shorted technology, because if you remember, it had a bridge amplification step where the reads were amplified to form a clonal cluster, to get the appropriate signal. So here it is no signal as such. A signal is basically and there is no DNA polymerase needed, right? Quick sample preparation, and basically the output is FastQ or FastFi file, which we will see today, right? A sequencer could be MinION, GridION, and for example, MinION may have about 2000 ports, but when you do the check, you may end up having like 1600 or something pores, right? And so when you start the run, the pore number drops. Then again it regains some number of pores, right? and in the next MUX scan, right? And when you see that okay, I have got sufficient data that I need, you can stop the run and you can wash the flow cell using the consumables provided by them, washing kit, and then again you can load another sample, right? So it is better that you may go for like a different library of the same sample, right? Because there could be some remnant DNA. This is basically suggested, but otherwise if you do the wash cleanly, I think there should be no remnants, right? And you may recover some number of pores. So basically the active pores are divided into 4 MUX, and right, and you can also allow like how much time you want to run it, okay, and after that the run will stop automatically, and you can also stop in between, right? So these are the different type of instruments available. So this is the USB based sequencer. You can connect to your USB port, okay, and your system should have 1 TB of hard disk free, and about 16 GB of RAM, right? And basically Core i5 or Core i7 above, right? And you can load your sample and get the data in real time. Okay, you can also do the base calling while it is going on, or you can hold the data, create the FastFi file, and then go for the base file to create the FastQ file, right? Similarly, there could be like this kind. This basically is replaced this by this one, okay, where you don't need a laptop and everything happens in this instrument. And there could be bigger versions like Gridan and Promethean also, which are for the bigger center like this, which can generate about terabytes of data, okay, but this can generate about 30 GB of data. But in real time you can see that you can get up to like depends upon your library preparation and something that unnecessarily blocked your pores, and these kind of things may happen. But like you can easily get like up to 10 GB for sure, and basically then if everything is right with your library, or you can also go up to 20 to 25 GB, okay. But so far in my experience also I have never reached to 30 GB. It could be their ideal conditions, right? And similarly for this MinION MK1C device, okay, so these are the different but the difference between these is that this one or that the tab based type of device MK1C, it is only one flow cell is used there, whereas in this one you can put 5 flow cell and this 48 flow cell. That means you can run like because every run takes like 24 to 48 hours, so you can run multiple samples together if it is a very high throughput lab or you want too much data. Okay, so this is the sequencer where you will load your data, and this is the flow cell, and this is the instrument which is reusable, right? And flow cell has to be discarded once the ports become 0 or less than usable, because library preparation cost is also considerable. So it is not that if you have like very less number of port, 100 or 200 left, you should go for a new library preparation, because that may not be worth spending a library reaction to go only for like limited amount of sequencing, okay. So as I mentioned, basically DNA, single stranded DNA passes through this, it blocks the ion current through this, and that blockage basically leads to disruption in the current which is measured, and that tells that which nucleotide has recently passed through this, okay. So like this, okay, it is detecting and the current is red, and that is translated into what nucleotide has passed through that, right? And because of this mechanism, basically it may have chances of errors, because you have to detect this current accurately, although their technology is also improving and errors are also going down, okay. So okay, again another picture of this, the same thing, that which is the disruption of the current detected when an analyte is passed, and analyte is here in this case the ATGC, which are attached, which are basically part of the single stranded DNA which is passing through, right? So now in real time on your PC or the device that is attached to the flow cell, you can see this kind of picture, which clearly tells that how the run is progressing. So you can see that these are all the pores which were there, right? of which 142 are currently sequencing. Okay, 18 pores are available for sequencing, and they may get DNA, and they may also increase this number of 142. 13 pores are unavailable for sequencing, that means you can no longer use that, and 321 are inactive. That means they may be inactive because they will be used in the another mux scan. So they will become again available in the next mux scan. So basically mux scan timing and this thing you can also change accordingly, right? So in each mux scan basically the number of pores changes, and the DNA is assigned to another set of pores for sequencing. Okay, so again, like this is the duty graph of nanopore sequencing. So at this point the sequencing is actively going on. These ones are there which can potentially contribute to sequencing, and this one is inactive ones which are not sequencing right now but may be available in the next mux scan, and this one is those, the dark one, which are unavailable for sequencing and they cannot be revived. Okay, so final report you get, basically you are getting a real-time report. So whenever you click on the summary page, you can get this kind of report, that how much is the estimated basis so far, right? And data produced include all the files that are created, so not very relevant. This number is relevant, like it generated 3.84 GB. Okay, total number of reads are 592,000 and a little more. Okay, and estimated N50 is 13 KB. Generally it generates more than 10 KB N50. Okay, N50 we discussed that it is the midpoint, or the point that covers almost half of your data. So basically higher the N50 means that you have longer reads in your sample and the quality is better, right? So it tells that how much time has elapsed. So basically the run was set up for 120 hours, but the user stopped the run at 27 hours 41 minutes. Okay, run stopped by the user, which you can always do if you think that you have got sufficient data, or it is not going on well, or basically suddenly the number of ports are decreasing, or anything that is unwanted. So you can stop the run, okay, wash it and again reuse the flow cell later, right? So it also tells that what kind of flow cell was used, okay, flow cell ID and kit that was used to do the library preparation, and the compatibility between the flow cell and the kit is very important, and you should always see before buying that what kit and what flow cell will go hand in hand. So basically for the purchase thing, you can order a starter pack which may include a single or two flow cell, or up to like 6 flow cells and 6 reactions, 1 flow cell each, like that, and some washing consumables which they provide in a pack. So in that case the compatibility is not an issue, right? But if you have already leftover flow cell or the reaction, then you have to see that whether the new flow cell or the reaction kit that you are buying, the library preparation kit, are compatible or not. Okay, and it provides the data as fast5 output, okay, which you have to use for the base calling. Okay, so basically this tells that what is the N50, or basically the kind of reads you have generated. So you can see that most of your reads are in this region. Okay, that means up to 12.5 to 20 KB, right? So 4 to 20 KB most of the reads are there, but read like maximum it have gone up to like 64-65 KB. That is the longest read that you have got in your data, right? So it gives you an estimate of how much reads are generated and what is the average length of reads, and whether you have really got very long reads, or sometimes you may see that it is only left to this. That means like there were a lot of shorter fragments in your sample and they occupied the pores first and led to like shorter reads. And there is like one basically a pack that they provide to remove the shorter reads also while doing the library preparation, that you can use to enhance the longer reads. And also if you start with high molecular weight DNA and which is like unfragmented, so the chances of good library preparation is higher. Okay, so basically this picture also tells like what is the pore activity. So you can see that these green ones were active, these ones are not active right now but available and can be revived later. These are unavailable and basically this is basically out of range or low, which cannot be used, right? So, and you have to observe this pore activity also soon after, like up to like 10 to 15 minutes or 20 minutes after you have loaded the sample, because suddenly you may see good pore number, or suddenly you may see like fall in the pore numbers. Okay, so at that time you may take a call to stop the run and reload the library, or prepare another library, or change something in your library so that it runs well, because that means an error, and unnecessary you will be spending time and wasting your flow cell if you don't stop such kind of runs. Okay, so the output files before base calling are basically, it forms a fast5 folder where files are saved, right? And these are other files that you may need to analyze your run, but generally like this is a folder that is used for the base calling. So after the base calling, we got a folder called fastq where the base calls have happened, and now you have base call means that from the signal that it has got, it is calling the bases ATGC. Okay, and generally like it's a time consuming process. You may also decide to do this while your run is going on, right? Or you can to avoid extra processing at that point, you can do it that later also, and later on you should preferably use a GPU to do faster calling of these bases, right? So basically after the read is generated, raw signal is converted to base calls, and then you can use like various methods, overlap, layout consensus, OLC, or we will see some other methods also based on deburrowing that is used for long reads also, and then go for the assembly and then correction of reads, long reads, by using the shorter reads or polishing step. Okay, so regarding the assembler, the base calling, Guppy is one of the commonly used tool for the base calling, and which is also provided by ONT. Okay, so basically it can call the bases from your raw fast5 files, okay, and converts that into ATGC, this kind of signal. Okay, it's a neural network based base caller, and in addition to this, it may also do the filtering of your reads, low quality reads can remove, right? And also can clip the adapters, but still some may remain, and that you have to do by Porechop or other methods, right? And it takes the file in fast5 format and produce the fastq files as the output format. Okay, so it can be downloaded from the Oxford Nanopore community page. So you have to unzip this. Okay, this is the file, right? And you are downloaded from the Linux 64-bit architecture. Okay, and generally that is preferable because you are doing a computing job, right? And then once you have done this, you can run the command, which is Guppy base caller, the input path of the fast5 file that is generated by the sequencer, okay, and where you want to store the output, what flow cell you have used, the flow cell type, what kit you have used, SQK, LSK 109 in this case, how many number of GPUs you are going to use. You may be having a GPU which can support multiple threads. So basically you can specify how many threads you can do, and that can really increase the calling rate and decrease the total time which is consumed. Otherwise it may take 2-3 days or even more if you don't use a GPU. Okay, then to remove the adapters, okay, so basically in one picture we have also seen that there were adapters. For example, here adapter is being read, here the basically the actual read sequencing is going on. Okay, so sometimes there could be some leftover adapters in the reads, right? And you have to trim, although Guppy could not remove that. So that you can do by using the Porechop, okay, which is good for removing the Oxford Nanopore adapters, right? So what it does is it has already a database of adapters and looks for adapter at the terminals and also in between, like it could be a case of chimeric read, and remove all those kind of adapters. Okay, so it was published in Bioinformatics Advances, and it is a commonly used tool for removing the adapters from the ONT Nanopore data, right? So you can download it from GitHub, and we also covered that while we are doing the Linux, I think. So basically this will remove the adapters. Okay, so it can be downloaded from GitHub, which is the source for mostly all the programs, right, repository for that, and then you will install it. So get it from GitHub by using this command, the path of the command at the program at GitHub. Okay, go to the directory, then Python 3, it's a Python program, and say install, and then it is installed. So once there is no failure message or something like that, that means it is installed correctly. Then you can go for Porechop -h, that will display all the help options. Okay, and it will basically do 4 things. One is these 2 things are optional, you can say like either of these 2 or both, right. So first of all, it will match with the available adapters which are available in this database, known adapter that is used with the with the Nanopore, right. And if it finds an adapter reliably, then it will trim the adapters from the ends, right. Generally it expects adapters at the end, but sometimes it could be there in between the read also, which could be a case of chimeric read. So now you have 2 options. One is to either remove the adapter, split the read into 2 parts, because you want to use the data, right, and you don't want to like lose data because of in between adapters, okay, or because it's a long read, and basically long reads, it could be one piece in your total DNA, and it may fit that position and help you to do better coverage. But okay, sometime you could be like don't want to compromise with quality and go only for the clean reads, then you can remove the option, do the option of discarding these kind of internal adapters, right. So command line is easy. So basically Porechop -i input file name, and then the output file name, and -o, okay, wherever you want to store, you can give a path or the output file name, and basically verbosity 2, that means you want to have the full summary of what is going on during the process, right. It will print on your screen, right. If you say 0, it will print nothing on your screen, the program will run and you will get the result, okay. And the number of threads you want to specify, and that is the beauty of Linux and power of Linux, okay, or GPUs, that you can specify that how many threads you want to use it, and that will enable like faster processing, and you can use the power of your system better, okay. So this is the file that is being generated, and it's run in verbose 2 mode. So it is displaying that start and end where the adapters were found, and they will be trimmed, okay. Okay, now you have done the base calling using Guppy and remove the adapters using Porechop. Now it is time to do the assembly, okay. So for which one of the best sequence assembler or commonly used assembler, which we also found good for our work, is Flye, okay. So it's a assembler that uses a repeat graph, which is helpful in resolving the repeat. Otherwise it uses the OLC method, overlap, layout, and consensus, right. And it is published in a Nature Biotechnology, a very good journal, and also had a huge number of citations, okay. So basically it's a de novo assembler, not a reference base. That means it is building the contigs and scaffolds from scratch, okay, from the Nanopore reads, right. It can take both Nanopore or PacBio data, okay, and basically it uses a repeat graphs as the core data structure that we will see in the next slide, okay. So basically it takes all the reads, see the align them and see that whether they are having some overlap, and if there is overlap between multiple reads, then it will kind of combine all those reads together and collapse it and form a single read from those, okay, looking at the consensus from all those. So basically this is called overlap, layout, and consensus part, okay. And also make a repeat graph so that it can resolve the repeats well, okay. So this is the situation, like this was the genome, and the repeats that it got from there. So these are the reads basically it got from there. So these are the reads that it got from Nanopore after running that Nanopore run. So basically you can see that there is overlap, you can see that this is overlapping with this and like that, okay. So you can go for the so if you align place the overlap reads like this, that is preparing the layout or overlap layout consensus, right. So you can see that basically this part is common between this part is common between okay. So you can merge all of these together to form a consensus sequence, right. and also you can go for, like, if you found some clues or evidences for this, you can create the concatenated disjointics. Okay. Now for the repeat resolving, you can see that it is doing all versus all alignment. These are the different type of reads, A, B, Y. Okay. So it is doing all versus all. So those which are showing some repeats, we are aligned here, are shown here, and otherwise it is mapping A versus A, B versus B, which will fall on this line, right? So this, using this repeat graph, it will resolve the repeats and then connect them together to form the correct assembly without much of the misalignments. Okay, so this program can be downloaded from this site, okay, from GitHub, and this is the program. Okay, and go to the directory. It is also written in Python setpy, and then install, and then basically it will be installed. And the usage is this, where there are several options, parameters you can define. Okay, and for example, this is the command line. So it is Flye -t threads, how many threads you want to run. So if your system has like 100 threads possible, then you can use 90 of them, so that 10 can do the basal jobs that a system should do, that may be kept free, and 90 you can assign, or in this case 40 is assigned here. The estimated genome size you may mention here if you know, or if you have some idea, right? Or then basically like Nanopore raw reads you are giving for this, so raw, and then you can see that this symbol we have used for the first time. This is used backslash. Generally we use the forward slash for the directory structures, but backslash only when your command line or the path is too long, and basically it doesn't fit in a single line, and you cannot press enter also to go to the next line because you are running the command continuously. So what you can do here is, you can put this backslash and then type the remaining command in the next line. So it allows you to go to the next line also. So this is the path to the Porechop FASTA file. So Guppy gave you FASTA file, Porechop remove the adapters. Now you use that as the input and specify the output directory of the Flye assembly. Okay, where your assembled contigs and sequences will be placed, right? So basically the output, it gives like these output files, of which assembly.fasta is the file that will be mainly useful for you, that contains the contigs and possible scaffolds that it has formed, okay, in the FASTA format, right? Now the summary of the log that it has created is like printed here. You can see that this was the command that you run, that okay, T68 total threads, genome size, okay, Nanopore raw, the path and the output directory, okay, then basically total length it has generated, genome size, okay, estimated coverage, that means average number of coverage of your reads, okay, 31 means that 31x coverage was average kind of coverage. Minimum overlap between the reads is 2000, that means when you are doing the alignment, you can specify this. It could be 1 KB, it could be 2 KB or 5 KB also, that if you have sufficient data, but 2 KB is a good overlap, okay, that you have kept for the between the reads, right? And the other parameters that were assigned. So this is the assembly.fasta file that it created, the contig number, the sequence, and the in the FASTA format, okay, and you can count that how many sequences or contigs you have got by grep -c, that means count this symbol which is present in the front of your read, okay, or basically the contig, right, a FASTA format indicator. So count this symbol from this file, it will give you the number of contigs that are assembled and placed in your file, okay. So this is the assembly info.txt file, which gives you summary of the contig number, the length of the contig, its net coverage or average coverage 33 KB, okay, and like other parameters from the graph. So basically this part is more useful for you, okay. So now Nanopore data we have seen, basically Flye we have also discussed, which is typically used for the ONT Nanopore data, but it can also be used for the PacBio data, okay. But so just to give you a quick overview or to refresh the PacBio SMRT technique that we discussed earlier. So basically The idea was from the double stranded DNA, basically you will be ending up like this when you are breaking the DNA. Okay, you are attaching the hairpin kind of adapters at both the ends like this. Okay, and then opening this DNA up, right? Then it becomes single stranded, and then the polymerase can read the DNA and create a copy of it. So this way, basically this can be done in a SMRT cell, or you can say ZMW SMRT cell, and this can basically create a copy, or basically while it is going through that making copy of it, you can read that what it is amplifying. So basically it is amplification free, and also at the runtime you can detect that what base pairs it is sequencing. Okay, so as the nucleotide is incorporated with the polymerase, the camera records the emitted light based on the nucleotide that is being incorporated. And good thing about it is that all 4 DNTPs are available at the time of incorporation, so that removes the bias of putting only one DNTP. For example, I put A, and basically G also, which is also a purine, have the like similar structure. Okay, and 2 rings. So basically G, if A is not there, G may take place of A, so that leads to error basically in case of some technologies. So here if all 4 are available in DNTPs, like in the case of a normal DNA replication in the cell, the chances of errors are less. Okay, so basically it uses a modified polymerase, and that emits the light when it incorporates a base, right? So again, like this was your DNA double stranded, you attached hairpin on both ends, and opens it up, and then use a polymerase to basically make a copy of that single stranded DNA, and while it is doing, you will be reading that what it has incorporated using the emitted light. So basically it produces a long read, very good for genome assembly. Nowadays it is like PacBio Hi-Fi is also available, which is having a just 0.1% error rate. Okay, can be very good for transcriptome, because you can get the complete RNA transcripts, right? And you can easily get information about the isoforms, and you don't need or get complicated due to the assembly of reads, smaller reads into RNA transcripts, which may cause issues. So very good for transcriptomics or epigenetics, because it can capture the methylation status also of the cell, right, of the DNA. Okay, and gives you a good uniform coverage. It is not basically biased by the GC rich or this region, which may happen due to the amplification step. Okay, highly accuracy these days it is improving, right? And single molecule resolution is also there, but the cost may be higher at this point of time. The instrument is also expensive. So for using this, mostly you have to outsource your sample, and because buying the instrument worth like more than 6 crores and is not possible over every lab, so you can outsource it. But still, for like every run may cost something like more than 5 lakhs, so it is a bit expensive. Okay, so comparison between the two, in this case basically polymerase is involved, 4 DNTPs are there. It is making a kind of circular structure of the DNA insert and the hairpins, and then using the polymerase to do the calling of the bases that it is incorporating by using the fluorescence, right? And in this case ONT, there is no polymerase involved. Okay, it is DNA which is passing through a pore and leading to a disruption in the current, okay, because of the relative size of the 4 nucleotides, and then you're using that information for calling which base pair it was. Okay, so theoretically it looks much cheaper, because no polymerase, basically no enzyme, no DNTPs. Okay, but accuracy could be an issue, and sometimes there could be basically blockers of pores if your DNA is of not good quality. Okay, or there could be some contaminants in your sample because you are extracting the DNA from basically plants or some insects or birds, which are typical causes of like blocking of these pores unnecessarily. Otherwise this is cheaper and can be used in your lab and can give you good amount of sequence data per run. Okay, so now wtdbg TDBG2 is also one of the sequencer that can be used, assembler that can be used to assemble the Nanopore or PacBio reads. Okay, So it assembles the raw reads without error correction and then builds a consensus from the intermediate output. So basically it chops the read into 1024 base pair segments. So it is using a fuzzy De Bruijn graph approach. So in the previous one it was using the OLC based method, overlap layered consensus method. Here it is using breaking the reads into smaller pieces and again creating a graph and resolving the graph to build the consensus again and building the your quantity, right. So basically the advantage of this one is that over the previous one is that here it may use lesser amount of computing power of your system, and you can use it if you have limited resources in terms of the workstation or the HPC that you are using, right. So this was also published in Nature Methods and also commonly used, but Flye I think is more used by people, right. But it may be like 2 to 17 times faster as compared to the other tools and giving almost similar contiguity and accuracy. And many times it could be, I'm telling you like multiple methods because it could be that you may like a particular method or you may try different methods on your data and whichever gives you a better assembly in terms of the assembly quality that you have checked, okay, and covering the genes, contiguity and coverage of the genome, you can select that method as the final method to report. Okay, so basically it is breaking the reads into k-mers and then of about 1 KB size, and then again you constructing a graph of those k-mers like we have seen in the De Bruijn graph approach, and then resolving the quantity, right, and creating the consensus from the graph. Okay. So installation again you can get it from GitHub and okay, the folder, and go to the folder, do the make, right, and then you can write this command. So it has 2 steps. One is like /wtdbg, one is x, that means you are using the ONT data, okay. Genome size is about like 4.6 MB in this case. Input file, total number of threads that you want to use, and the output file, okay, with basically the extension as dbg, okay. So basically this is the first step, and then to get the actual consensus you will have to use the next command. So for the first wtdbg or de novo assembler, okay, for that these are the options which are available, okay. I have told you like standard command that is used, but you can also try other options to fine tune or improve the basically the assembly. In this second step, when you are building the consensus, so the command changes a little bit, okay. This is the command and number of threads, input file that you got from the previous step, and the output file, that is the dbg raw.fa, that means the FASTA file or the assembled config file, okay. And these are the other parameters that you can use with this command wtdbg cns, right. So basically there are so many options available to fine tune your assembly, but going with default may be okay, right. And if you have like sufficient knowledge of the system, you can also define k-mer size and other things that you want to use for this, right. K-mer size in case of dbg, right. So this is the output of the first step, right, of ctglay.gz file, okay, which is having more information about the raw reads and the config layout and edges it formed in the first. And the second step, it created the contigs or the final consensus of FASTA files, okay, which is like config to length this much, okay, and about 1 MB or more, right. So this is the final config file. Okay, the third assembler is SMARTdenovo, which is good for long noisy reads, basically the Nanopore or PacBio reads, right. And it can produce like reconstruction of reference quality genomes, de novo assembly, right. And it uses an overlap layout consensus algorithm that is used by the Flye also, which is commonly used by programs to assemble the longer reads, okay. So it's a de novo assembler, and it does the all versus all alignments, and then based on looking at the overlap between the reads, okay, it places the read in that order, overlap layout, basically lay the reads in that order and build the consensus for that. Okay, so it has there could be like 4 different command line tools for this, okay, to do the read layer overlapping, okay, rescue the missing overlaps and identify the low quality regions in camera, and then to produce the final unitig or the consensus. Okay, so again this can be got from the GitHub site, and once you have downloaded it, you can do the cd, go to the directory and do the make, which will install it for your system. Then usage is like you can say SMARTdenovo.perl and PL, and the option that you can give from here, okay, or with the various arguments. So basically for making the raw unitics, the command line could be SMARTdenovo.pl and C1, okay, that means generate the consensus 1. The generate the consensus, the read input file is read.fa, and this is the output file, and on this output file you will do the make -f and the output file name that you generated from here, right, which will build the final unitics for you. Okay, unitics are pre-contigs, or basically which can be converted into contigs later, right, and so this is unitig 0 length, and it's the node from which it is got. Okay, so basically there is a variety of assemblers available. I have given you some of the common ones, and among which I think Flye could be a good option, and wtdbg2 is a good option for like if you have like limited RAM and CPUs, but still I think the performance of Flye was better in our opinion, right. So now you can compare like different assemblers, and you can use that for your purpose. So sometimes it could be that Flye may fail, but the other one will do the job, and then whichever suits you or gives you a better output is the all are published in good papers in good journals, so all are reliable and well tested, validated. So depends upon your choice, and you may try more and use the one that is better for you, and all are also evolving based on the reports by the users on the various review forums, and where you can see that the errors that you may find or their solutions and how to do the assembly better. So you may also like to read those user forums to get more details about this. Okay, so it is also depends upon how you are using a tool. Okay, I may be using one tool better and thinking that that is performing better, okay, but the other tool I may not have used that well. So sometimes it's usage, and reading these review forums also helps, right. So just to compare, like if you run it multiple times, so basically if you by different assemblers, so in the case of say Amla Genome, right. So if you do the assembly by like wtdbg2 for the Amla Genome, you can see that it gave a very good N50 of or maybe the highest among all of 314 KB, right. But the actual genome size which was estimated was something like 579 MB, but it gave you a genome size of 433 MB, right. So it was lower in terms of the total coverage of the genome, right. And then when you use another one, SMARTdenovo assembler on the same genome the data, okay, you can see that it improved the genome size from 433 to 465 in this case, but still was lower than the estimated genome size. That means coverage, for example, if you give coverage as more importance compared to N50, so N50 also was lesser than the previous case, right, and both of them predicted the GC 33% and 33% almost correct, right. So okay, for Flye basically you can see that the N50 was the lowest among all, right, but still it is not bad. It is like 267 KB, right, but it could reach to like 528 MB of the total coverage of the genome, which was estimated to be 579 MB. Okay, so this estimation also gives you a kind of idea of like how much coverage you expect from the assembly, right, but it is not that this number will be correct. all the time. Okay, so it is just an estimate, and basically higher quality assemblies or chromosome level assemblies or more data will definitely reach you to a better number, or like more correct number in this case, right? But still, like, since you have estimated one number, you may prefer to select an assembly which is more nearer to that estimated assembly size, right? So like for example, in case of turmeric genome, you can see that 41x nanopore data was used, and the N50 was about 100 kb, and Busco was 92%, which is kind of reasonable. For the banyan tree, you can see that the N50 was higher, like about 480 or 500 kb, and Busco was also like more than 95%, which is good. Okay, again, like in this case, the N50 was 179 kb in case of Jamun, whereas Busco was more than 98%. That means almost 98% genes you have covered, which are single copy ortholog SCOs, right? Which gives you like an idea of that your genome is kind of complete, right? And N50 is also good enough to go further, right? In case of Amla, you got a very good N50 of 597 kb and also Busco of more than 98%, which means that it is good quality assembly for going further for the chromosome level assembly, but anything above 0.5 MB or 500 kb is good enough. Okay. So basically in this lecture we looked at the long reads sequencing technologies and their assembly methods, the common ones, the advantage of using different methods and different parameters, where to download, how to prepare the data for assembly, okay, base calling, and also the removing the adapter. So basically this lecture provided you an overall summary of how you will proceed for the long read assembly using either ONT or PacBio data. Okay, so for try, you can also download the data from available websites like NCBI. From any published genome, you can download their data and do the assembly and try on your system just to learn this assembly, or you can also create or use on your assembly. So so basically for this lecture we are good for today, and thank you.